in the world of proteins, proteomics, is the driving force of the company towards developing affordable biotherapeutics. Mr. Narasimha Rao Nedunuri is the managing director of Clonz. Narasimhaji is one of the key founding members of the company. Narasimhaji's research experience in cancer biology at CCMB, Center for Cellular and Molecular Biology, helped him to contribute through his work at a USA based MNC as product manager for its Indian subsidiary and also as a group leader. Proteomics and genomics for a reputed CRO prior to the birth of clones. His wide exposure to countries like Malaysia, Korea, Thailand, China, France, Germany, Switzerland, and USA on various business assignments is also helping in positioning the company. Ladies and gentlemen, please join me in welcoming Mr. Narasimha Rao ji on stage to address us on monoclonal antibodies for infectious diseases, a paradigm shift. Mr. Narasimha Rao Nedunuri. Nedunuri.
This is forward, this back. Yeah, okay. Thank you. Very good morning, everyone. First of all, thank you, Mr. Satya, and for inviting me here to talk on monoclonal antibodies. Well, many of you, being from pharma industry, may be knowing about monoclonal antibodies. They are the most sought after and uh, the drugs that every pharma would like to add on to their kitty because of the certain advantages that drugs have, precisely working on specific targets to uh, offer the benefit without having major side effects or interfering with the housekeeping mechanism. So these monoclonal antibodies are very expensive affair. They're not only the cost, they're not only the complex ones, molecules, but also uh, uh, very time taking to develop, capital intense, and also what I can say high dividend as well, but provided you get there with a success. So today I'm here to talk about monoclonal antibodies, which we call as MAbs shortly, for infectious diseases. There's a shift that we are looking at now with COVID-19. So let's look at these MAbs, how they started and where we are today and where we are heading in terms of the infectious diseases. But prior to that, I'd like to introduce, these are the contents of my presentation. Apologies, as this may not be a very colorful presentation. I made these slides a few hours ago. I just do the introduction and then talk about the beginning of monoclonal antibodies, then MAbs to treat bacterial infections, followed by MAbs to treat viral infections. Then again, talk about MAbs for infectious diseases, pros and cons. Then finally, I end with what can we expect in the years to come. Time given is 15 minutes. I'll try to be as quick as I can. Just to brief about myself, as I was introduced already, very briefly, a molecular biologist turned entrepreneur. So one of the founding members for the company called Clonz Biotech, which is almost a decade-old company. Not many of you might have heard about us. We are still in the gestation period. Now you know how difficult to bring these products into the market and how time-taking they are. So we are, I did research in molecular oncology, proteomics at CCMB, then moved to a US-based company. I'm also one of the mentors at CCMB Incubation Center, ICIA, IIT Hyderabad. Then also the committee member at TS Cost of Telangana, Science and Technology. Then member of CIA Biotech Committee. Then I'm also part of a coordinator at Chris Mason's Weill Cornell Medical Center's Global Metagenomic Project. I'll just here, I stop and share my experience here. 5 years back when we initiated the Indian metagenomic study analysis, where we take samples from subways or maybe what we can say, the metro stations basically. So we take the surface of the commonly accessed areas and take the DNA out of it and then study what microbes are going in there. Then not many people were keen at that point of time. We tried to explain them that this is very important to know the surroundings and the bacteria and viruses we live with. There was a poor response. Still, we could start the study. Now we know how important. People are spending money now to swab their surroundings and see what microbes are there. So at times we lack in the vision, which we have to pay price eventually. So just to briefly talk about Clonz, we are a clinical stage biopharmaceutical company promoted by the young group of passionate biotechnologists focused on development of therapeutic biosimilar monoclonal antibodies. My colleagues are here, Mr. Murli, Dr. Kara, Vijay. So we started this company in 2010 to produce these biosimilar monoclonal antibodies. We are stationed at Genome Valley, Hyderabad. So in a biotech facility built by Department of Biotechnology, it was an incubation center. Later we progressed to a bigger laboratory close by. So our core competency is mammalian-based biosimilar monoclonal antibodies development. Probably I'm proud to say that we are the only clinical stage startup biosimilar exclusive monoclonal antibody company of this country. Now quickly move on to the topic. The beginning of monoclonal antibodies, which as a layman we know that antibodies are produced within our body. They give us the protection against the foreign invaders like bacteria, viruses, anything. But here, the monoclonal antibodies are the ones that we make. They are man-made proteins that are identical and act like human antibodies binding to specific targets that we intend to do. So in 1975, Cesar Milstein and then George Kohler, they developed the technique for making monoclonal antibodies, and they won the Nobel Prize in 1982. So those are the ones who made this happen for the first time. And ever since this happened, there was more research went in to develop these monoclonal antibodies. And these antibody-based drugs have fewer side effects, as I mentioned earlier, due to their high specificity. As a result, the therapeutic antibodies have become the predominant class of new drugs developed in recent years, and they are the most sought-after drugs And we will slowly move on to COVID situation and see where we stand in terms of the monoclonal antibodies there. The majority of the monoclonal antibodies are used to treat cancers and majority of the autoimmune diseases. So they are predominantly there in that domain. However, the emergence of antibiotic resistance and recent pandemics, they are driving the development of MAbs against these infectious diseases. If you look at holistically, in a nutshell, uh, the infectious diseases we know they're caused due to parasites, viruses, bacteria. If we consider them as antigens, the monoclonal antibodies are targeted against either neutralizing those pathogens directly or targeting their entry points to check them off. So then these monoclonal antibodies can be used either to have a therapeutic agent or to have vaccines or to have diagnostics. We get the test done, but we don't know that behind every test that we do for the antibodies, these antibodies are, the monoclonal antibodies have to be there to do the test. If you look at antibodies to target the bacterial infections, we know that antibiotics are there. Why antibodies? Monoclonal antibodies. So antibiotics over a period of time, we know that most of the pathogens or bacteria, they develop resistance over majority of the antibiotics that we use. And it has become a global problem now. So indiscriminate use of antibiotics and in fact microbes are also getting clever. So they're becoming more intelligent. So they're becoming adaptive to these drugs that we use, and it's becoming very difficult to treat them. And globally, this burden is huge. There are more deaths today due to infectious diseases than anything else. Of course, we need to have alternative methodologies to look at controlling these pathogens. If you look at, there are ways that either we can directly target those pathogens or like target their molecules to survive and replicate, like neuraminidase and then hemagglutinins and things like that. Antibiotic conjugates are very interesting concepts that are being developed nowadays, and it's been one of the target areas of many companies. If you look at this, you may be very interesting to know that some of the microbes, they go inside the cell and live. Antibiotics will go and antibodies will be only for those who are external to the cell. It's very difficult for some of those the antibodies to go inside the cell and have this thing. So it's what the people are looking at, the scientists are working at clubbing the antibiotic to an antibody and making it travel inside the cell and getting cleaved inside, and both it does the act of antibody as well as the antibiotic. So this is a more effective way. So far, we're very limited successes, but we are going to be there soon in this domain. If you look at antibacterial monoclonal antibodies, they offer new ways of fighting antibiotic resistance and control those bacterial infections. If you look at some of the examples here, the toxins that are there, it has become a very what I can say, the favorite areas to be looking at, several targets of these bacterial and uh, viral products. For example, this toxins, toxin B, that actuximab, it binds this toxin, reduces the burden basically, and we know uh, septicemia is becoming a very, multiple uh, pathogens taking over our immune system and complete control, uncontrolled growth. Some of the toxins that if we bind them, so they will not, they will reduce the burden and slowly there is an improvement and this thing. So several toxins, we have monoclonal antibodies, as we can see. Um, like anthrax, we have monoclonal antibody against it. So some of them are clinical trials, some of them are now in active research. If you look at MAbs to treat infectious diseases that are specifically the viruses, no, that's the need of the hour today. The whole world is now struggling with how to contain this virus, SARS-CoV-2 specifically. Now we had their sisters and brothers coming in some time ago, disturbing us to some extent, but we were able to contain it maybe because their R naught is not that great, and we are we were very careful, or we were not too traveling that time across the globe. So we were fortunate to contain them, but SARS-CoV-2, we've seen how quickly and rapidly it spread across the world. And so this, before even COVID, there were several attempts to develop some of the monoclonal antibodies against some of the viruses, a few viruses that we felt, for example, Ebola. So we had successfully developed some of these, and then we also had developed MAbs for syncytial virus, the respiratory syncytial virus. That has become a successful one. Then we have against corona, we have several MAbs that are coming up. Now, what I personally saw, several people who took monoclonal antibodies, that cocktail, they quickly recovered in a couple of days. Those who are elderly people, those who are susceptible and more, you know, diseases burden they have, and those people really, the monoclonal antibody cocktail helped to a great extent. Why? Because these are highly specific antibodies, they go bind to them. But again, when we say highly specific, the question needs to be asked, what if the virus changes its conformation? And well, that that ambiguities continuously exist as long as these viruses are in existence, because their fundamental nature is to change and evolve and mutate. But fortunately, most of the antibodies, monoclonal antibodies, they were planned such a way that at least some of the conserved sequences, conserved epitopes, what we talk, so they will be covered. So so no matter we have minute mutations, they will still be captured or neutralized. They will be bound. So we can see with the emergence of even several variants, but the best part is our own immune system. As you may be aware, that if a vaccine confers immunity by binding to, say, 2 or 3 sites on the virus, if we develop our own immune system, so our immune system maps it so well that not just one or two, but hundreds of epitopes are at a time targeted, and we have a perfect and excellent immunity across these viruses that we exposed to. Because our immune system captures most of the conserved areas as well, which usually don't get changed. But of course, we cannot be so precise and intelligent as our own body, but we can do to some extent arrest these viruses by targeting few key areas. Like we can two ways we can work it at. We can check their entry points, target the monoclonal antibodies at their entry points, and then stop them there, or we can target the virus itself so that the monoclonal antibody goes and binds on a site and gets neutralized. So briefly to talk about MAbs for the pros and cons of these. Uh, Pros, of course, they are safe as they do not interfere housekeeping and are highly specific. So they don't do much of a harm that chemically synthesized drugs do, but functions quickly, unlike vaccines. Vaccines, we know that we need to spend so much of time to develop them and ensure their safety and efficacy. However, when compared to those vaccines, monoclonal antibodies are quick, uh, then also quicker to develop than vaccines. So not only the regulatory pathway, but also how we develop them and manufacture them. But can be a solution for multi-drug resistance. So for bacterial infections, as we heard, we have seen it before. When it comes to certain shortcomings and cons, as we said, trial design to lethal diseases is very challenging here. The challenging against biofilms and intracellular bacteria. So that's quite challenging here. We know bacteria, if we are able to reach through our blood circulation to the areas wherever we can go, we can effectively arrest those replications of these pathogens. But what if they are away from the blood circulation, inside the cell or biofilms? It's a thin cover coated these things. They live in this bacteria inside our body. It's very difficult to reach those places. So that's quite challenging for bulkier molecules like monoclonal antibodies. It's not easy for them to travel to those places and give us the protection. Well, these are very expensive drugs. As you know, the existing drugs that we have, the monoclonal antibodies we have, I think it costs us around 1,20,000 a twin pack. So for two people, so that's quite expensive. That's quite understandable because the technology itself is expensive in this domain. Of course, the innovation costs. The innovator has to recover their innovation and the cost that they will be spending in. So usually they are high, but given the situation like this, they are lifesavers. So we see these MAbs are going to explode in the years to come because of those reasons what we just spoke. If you look at what we can expect briefly in the years to come, of course I can list a lot of things, but very briefly, the challenges that we have are like immune evasion. Then we see T cell exhaustion that happens. We know repeated exposure of antigens to the body, the immune system. Our own T cells get exhausted and they don't respond well. So that's one challenge. And then multi-drug resistance that is happening. Then also pandemics like this. They are the key drivers of this innovation. The last slide of it. Oh, there. I can see the presentation this way. There. So all these challenges are driving this innovation towards MAbs-based applications to control these infections. That's going to be the favorite area for the scientists to be in. To take up those challenges. We as a scientists were very challenged by this virus, and we all, any scientist cannot rest peacefully if the challenges are thrown. So, as biologists, we have been also looking at several ways to work towards developing therapies or solutions for this. In fact, we in the beginning we tried and we identified 9 targets to develop monoclonal antibodies against these viruses. But as you know, we are limited with funds. We are a startup. So we tried to get funding from the government, which we couldn't, unless the government funding is there for this. We cannot even think of developing these molecules. Such an expensive affair it is. So of course, these are the challenges. What I mean to say is these challenges really, ignite the minds of the scientists to even take up, work maybe out of the box and find solutions. Our team is also continuously working on these things, but of course, this is a new thing came up, and it's not our forte, or it was not in our bucket list. So, and also the funding requirements and all, we kept it aside. But of course, globally, this is going to drive, these challenges are going to drive very, very innovative therapy options that we may see in coming years. Then antibody engineering to have T-cell engagement, multivalent, multi-domain antibodies. So, and also the delivery systems. We are going to see some very, very innovative delivery systems even for monoclonal antibodies. As you know, monoclonal antibodies are mostly by IV infusions into the blood. But we can see some novel transdermal applications, even oral as well as needle-free administrations. They're going to come up down the line. And MAbs that can work partly as therapeutic agent and partly as a vaccine agent. It's very interesting. A monoclonal antibody can be a therapeutic agent as well as, it offers immunity. It works in between. It triggers the immune response, and it is stored in the memory as well. So those are going to be the areas which are going to come up. But of course, for any of these things, collaborative efforts between hospitals, physicians, academia, scientists, and the industry. Most importantly, industry, because it's an urge to develop solutions to a problem, because they all already have an equipped solution for some of the diseases. So industry cannot be ignored here, but of course the coordination required between the doctors and scientists, also academia, because without the fundamental science, no innovation can be built. So all these collaborations have to happen. Then of course, the fundamental key factor apart from all these things is the funding. The the money is required to augment the innovations for addressing these challenges of pandemics and all these lethal diseases. And they, they are in, they're expected to grow further as we came here. So there is no other way to contain these except, um, you know, with the innovation. As we know, everybody started asking, where is the vaccine? Why the vaccine is not coming up? So why the solutions are not coming up? They will not come up until the minds are ignited and they are supported by the systems early on. Um, then the whole ecosystem have to be there to have these solutions coming in to rescue all of us. And these are going to come irrespective of whatever it is. The challenges are thrown to us are very big and very disturbing to many of us. So innovations do come. The only good thing about this pandemic is the ignited minds. They are more now. People are challenged and people are looking at these innovations in a very, very aggressive manner. So what I can say. So I wish we all have that ecosystem building in our country to bring more affordable as well as specific and significant solutions coming in. So with this, I end my presentation. Thank you very much.
Thank you, Mr. Narasimha Rao ji. I would request you to come back on the stage, as I also request Mr. Satya Brahma ji to come on to the stage to present a memento as a mark of respect to Mr. Narasimha Rao ji.
He spoke about innovations here. Exactly, that is what I was speaking in the morning session actually. So government needs to come forward. How many rounds you have made to the government of India for presentations? They must have promised very rosy picture to you, but they didn't come back to you.
No, I cannot be as aggressive as you.
No, I'm a journalist, I can speak. No, I also want to know one thing. During my last talk with you, you have tied up with a major pharma company. Would you like to disclose that name?
Yeah, it's Sri Krishna Pharma, which is a 4 decades old pharma company in India. It's the fourth, it's well known.